Hi, and welcome to our webinar, Genes to Proteins in Days, Accelerating Protein Expression and Purification with eProtein Discovery. My name is Lisi Su. I'm a Senior Commercial Product Manager for Synthetic Biology at Integrated DNA Technologies, and I will be serving as the moderator for today's presentation. My team supports products spanning from fragments, genes, vector onboarding, and engineering. We recently launched our SynBio Custom Solutions team to focus on innovation alongside our customers and drive the development of products and services outside of our catalog offering to better support the evolving needs of scientists. At IDT, I lead product strategy for synthetic biology, supporting a wide range of applications including antibody discovery, protein engineering, and therapeutic development. Today's webinar will be presented by me, Lawrence Welch from Nuclera, and Joel Francis from Aldevron. Lawrence Welch is an inside sales specialist at Nuclera, where he supports the adoption of the company's automated cell-free protein expression system, eProtein Discovery. He earned his undergraduate degree in pharmacology with industrial placement experience at the University of Manchester and completed his PhD in Sean Moran's lab at the MRC Laboratory of Molecular Biology. Since transitioning to a commercial role in 2025, Lawrence has focused on helping researchers use cutting-edge microfluidic technology to accelerate protein discovery. Joel Francis is a senior scientist in the protein R&D group at Aldevron. Joel completed his bachelor's degree at the Grand Valley State University, where he majored in cell and molecular biology and minored in chemistry. He continued his training as a graduate student at Stanford University Department of Biology. His research revolved around the enzymology and functions of protein lysine methylation, gene expression, regulation, and cancer biology. Since joining Aldevron in 2023, his primary work revolves around the CRISPR gene editing product line. During today's webinar, please submit your questions using the questions box. Any questions that are not answered during the Q&A will be followed up with afterward. For those who are not familiar with IDT, we were founded in 1987. Our mission has always been to support and empower researchers, and we serve more than 130,000 researchers. We see our role as your partner, not just a provider. The 121,000 publications our customers have contributed to aren't just a number. They are a reflection of their breakthroughs, their innovations, and their impact. IDT serves as a market leader in DNA and RNA synthesis. We offer reliable, customized solutions and tools to provide actionable insights, elevating your work and simplifying your workflow at scale. Operating with vertical integrations across 11 countries around the world, we help you achieve what was previously considered impossible. IDT is proud to be part of Danaher. Danaher's science and technology leadership puts IDT solutions at the forefront of an industry so they can reach more people. Being part of Danaher means we can offer unparalleled breadth and depth of expertise and solutions to our customers. Together with Danaher's other businesses in biotechnology, diagnostics, and life sciences, we unlock the transformative potential of cutting-edge science and technology to accelerate the pace of research. IDT is a global leader in biosecurity and biosafety. We are a founding member of the International Gene Synthesis Consortium. And as part of our commitment to biosecurity, we conduct vigorous biosecurity screens. We also adhere to strict guidelines to ensure sequence information is secure and confidential at IDT. We are committed to shaping the future of sustainable science. Phosphoramidite chemistry, the method used to assemble oligonucleotides and employed to manufacture the majority of IDT products, produces hazardous waste. In Coralville, for over 15 years, IDT has pioneered new and innovative ways to reduce the amount of hazardous waste we generate. Initial efforts involve evaluating less hazardous solvents for all synthesis reagent solutions and switching to less hazardous materials when possible. Further evaluations allowed us to remove all non-hazardous aqueous material from the bulk hazardous waste stream in our Coralville labs. We also evaluated processes that created smaller waste streams to determine if we can make modifications to eliminate the hazardous waste created from them. As part of today's webinar, I'll be sharing a brief overview of our synthetic biology portfolio. IDT's synthetic biology portfolio offers solutions across many applications, ranging from qPCR controls, in vitro transcription, CRISPR editing, to recombinant proteins, and more. We do this through our high-quality synthetic DNA products with fast turnaround times and customizable features. Our E-Blocks, G-Blocks, and G-Block Hi-Fi are versatile double-stranded DNA fragments that can be used for gene assembly, antibody construction, or as controls and in other genomic assays. Our custom gene synthesis product offers NGS-verified sequence-perfect clones with your gene of interest cloned into your custom vector or our selection of catalog vectors. Last year, we launched our Rapid Gene product, which offers fast turnaround time for custom genes cloned into any catalog IDT vector in as little as 5 days. With all of our clonal products, there are no cloning fees. This year, we launched our SynBio Custom Solutions team to better support the evolving needs of scientists and to innovate alongside researchers to advance the pace of innovation. Our SynBio Custom Solutions team is a specialized team of scientists dedicated to partnering with researchers from concept to delivery on SynBio projects. We leverage IDT's years of experience to enhance the design, build, test cycle and remove the rigid requirements of high throughput manufacturing to provide highly customizable solutions to our partners. Please feel free to reach out to learn more. IDT offers a comprehensive product portfolio that supports synthetic biology. We continue to partner with researchers to create products that help drive innovation. Thank you for the opportunity to share a brief overview of our synthetic biology portfolio. Now I'll turn it over to Lawrence to talk about the complete Nuclera eProtein discovery workflow.
Thanks for that, Lucy. So now I'm going to talk to you about how with eProtein discovery, you can accelerate protein expression and go from genes to proteins in just days. So what you're looking at here is Nuclera's eProtein discovery system. It's a compact, walk-away benchtop system, and it combines digital microfluidics with cell-free protein synthesis to streamline the process of screening expression and purification conditions. The result? Researchers can quickly access high-quality, assay-ready proteins, accelerating their downstream experiments with minimal hands-on time. So, what's a problem we're looking to solve with eProtein discovery? And it's what we like to call the protein problem. And so this slide shows a traditional bacterial protein production workflow. And so imagine you're kicking off a new project. You start by designing your construct, cloning your gene into a plasmid, transforming that plasmid into your preferred bacterial strain, and then comes growing the culture, inducing expression, lysing the cells, and finally, multiple purification steps. But lo and behold, after all of that, your protein yield is low. So maybe it's crashed out of solution, or it's barely expressed, or maybe even worse, the quality's poor. You find that your SDS page gel has more bands than a Spotify playlist. And that's because maybe it's been contaminated by host chaperones, or it's been degraded by host cell proteases. So now you're back to square one, redesign, restart, repeat. This workflow is not only time consuming, but also requires highly skilled protein scientists, and it comes with a high failure rate. The result? Serial iteration that can drag your timeline out from weeks to months or even years just to obtain a usable protein. So, how does eProtein address the protein problem? It's built on 4 key pillars of technology. And first, we have digital microfluidics, shown in the video here. And what you're seeing are hundreds of nanoliter scale droplets being moved using electrical signals in a fully automated fashion. And I like to think of each droplet as, a miniature reaction vessel, and every movement replacing a manual task at the bench, say like a pipette stroke or a centrifugation step. The second key pillar of technology is cell-free protein synthesis, which powers the unique biochemical reactions inside all of those droplets. And this lets us bypass living cells entirely, giving us faster, more controllable expression. And so the third key pillar of technology is we leverage split GFP tech to measure not only protein yield, but also protein quality, using fluorescence to assess folding and solubility. And I'll cover that in a bit more detail later. And so last but not least, the final core pillar of technology is our eProtein Discovery Cloud software, which helps guide your experiment in an intelligent manner. Ultimately, what this means is that with eProtein Discovery, you can go from DNA to assay-ready protein in just 48 hours. That's a dramatic acceleration of the workflow, turning what once took weeks or months into just 2 days. And this unlocks faster progress in biochemical, biophysical, and structural biology studies, letting you move into downstream experimentation almost immediately. So now let's take a higher level look at the protein discovery workflow. And this process is divided into 3 key stages, all guided by the eProtein Discovery Cloud software, which you can access remotely anywhere in the world. So step 1 is the design and prep stage, where you design your constructs and expression conditions. And for the DNA design, you simply input your primary sequence and the software will take care of the rest. The second stage is the automated screening. So once you've prepped your DNA, you simply load them along with your different cell-free blends and magnetic beads onto the cartridge. This will then initiate a 2-part 24-hour screen, testing first 192 different expression conditions, followed by a targeted purification screen of the best hits. And then finally, from that screen, you then extract what I like to call the winning e-recipe, which are the optimal expression and purification conditions for your protein of interest. And these are then used the very next day to scale up your protein production, either on the benchtop using cell-free reagents or in a shake flask with E. coli. So now let's take a closer look at the different dimensions of screening. So first is dimension 1, shown in green here, and that's essentially the ability to look at different protein constructs. So you might be working on multiple different target proteins or testing a series of engineered variants. The next is dimension 2, shown here in blue, and that's for solubility tag screening. And so we offer a menu of 7 N-terminal solubility tags plus a no tag option, giving you 8 options in total. And finally, the third dimension applies across all workflows, which is expression condition screening. Our standard kits include a range of additives to boost protein expression, and these include things like cofactors and redox regulators. But look, we know protein expression isn't a one-size-fits-all, especially for challenging targets or new-to-nature proteins, and that's why we've introduced a custom additive feature which allows users to load in their own reagents, opening up powerful new screening capabilities. With this feature, the system adapts to your protein needs, not the other way around. The protein discovery system can work with 2 different types of DNA input, offering flexibility. So the first being linear e-genes, i.e. DNA fragments, or the second one being circular e-genes, i.e. plasmids. So first of all, starting with the linear input, we provide linear e-gene preparation kits that contain megaprimers designed to flank the linear G-block DNA fragments you receive from IDT. And these are then assembled into a fully functional expression cassette through a simple 1-step assembly PCR. Together, these megaprimers simplify construct generation and ensure that your DNA is fully compatible with our expression system. Look, and so we understand that not every lab has easy access to molecular biology workflows, and that's why we've introduced an alternative in circular e-genes. And so these are ready-made plasmids that can be ordered directly, eliminating the need for PCR assembly. It's a streamlined, plug-and-play option designed to make eProtein discovery even more accessible. So far I've talked about DNA preparation, but now let's move on to the expression conditions. And so at the heart of this is our cell-free core, which consists of recombinantly produced E. coli transcription and translation machinery required for protein synthesis. And this core can be supplemented with either Nuclera-provided additives or your own custom reagents, depending on the needs of your proteins. And once you've prepared your E-gene constructs and your cell-free blends, you're ready to start the screen. You simply load everything onto the digital microfluidics cartridge using a multi-channel pipette, and the system takes it from there, running a fully automated expression and purification workflow. It will screen 24 different E-genes against 8 different cell-free blends, giving you 192 unique expression conditions, with the top 30 expressors down selected for automated cartridge purification. So to assess both protein yield and quality, we use a fluorescent protein characterization assay based on split GFP. And so when a protein is well expressed and properly folded, as shown in the blue square, you get the result is a strong homogeneous fluorescent signal. But in contrast, if the protein is poorly behaved and it forms aggregates, you can see it crashing out of solution, as those aggregates will appear as fluorescent puncta, and our inbuilt AI analysis will automatically distinguish between soluble signal and aggregated signal. So after completing this 24-hour two-part screen, your results are available anywhere in the world through our eProtein Discovery cloud-based software. And so the plots I'm showing are very small, but what I want to emphasize is just how rich and comprehensive the data output is. And you get what I like to call this treasure trove of information. First of all, covering 192 expression points shown by the top panels, but also you get a detailed readout of purification performance as shown in the middle panels. And so by integrating both expression and purification data, the software doesn't just help you identify the winning recipe, it also enables you to predict scale-up yields for the very next day. And now let's talk about that scale-up reaction the next day. It uses the same DNA, it uses the same cell-free blends, and it uses the same magnetic affinity beads as on cartridge, but this time it's in standard centrifuge tubes, allowing you to produce microgram to milligram quantities of protein. By simply washing and eluting beads, you typically achieve about 90 to 95% purity, with no need for size exclusion chromatography or other complex purification steps. The result is high quality, assay-ready protein, fast and efficiently. And what's more, if you require more protein, protein yield is directly proportional to the scale-up reaction volume. And look, so for many projects, scaling up in cell-free works. It works perfectly, but in other cases, it might be more beneficial to scale up in bacterial systems, and that's where eProtein Discovery continues to support you, helping navigate the well-established path to cell. And that's because the optimal conditions identified during the on-cartridge screen can inform key parameters for bacterial expression, such as which strain to use or which construct designs to prioritize. And it's in this context, our circular E-gene constructs enable a seamless transition from cell-free screening to in vivo scale-up. So to conclude, I'd like to share a non-exhaustive list of soluble protein types that have been successfully produced using eProtein Discovery. And what's exciting is that this list doesn't just represent a variety of protein classes, it also spans proteins from across the kingdoms of biology. And finally, I'm pleased to share that eProtein Discovery now supports the production of membrane proteins in addition to soluble proteins. We've already demonstrated success across key membrane protein classes, including GPCRs, ion channels, and transporters. And so to finish, I'd like to summarize the key points. So firstly, with the eProtein Discovery system, users can go from DNA to assay-ready protein in just 48 hours. And this speed is enabled by an on-cartridge automated expression purification screen, which allows users to interrogate up to 192 expression conditions and 30 purification conditions per run. And it's from this screen the winning e-recipe can be taken forward to a cell-free scale-up on the bench, or users can opt for a seamless transition into an in vivo workflow using circular e-genes. And finally, all of this is facilitated by eProtein discovery software, enabling built-in detection, yield characterization, and result visualization. And so with that, I'd like to hand over to Joel from the Aldevron team to discuss a real-life example of a gene-to-protein workflow.
Thank you, Lawrence. As he says, my name is Joel Francis. I am a senior scientist here in the protein R&D group at Aldevron, and I'm excited today to be able to share with you some success that we've had with the eProtein discovery system. So the core cast of characters for this talk begins with proteins 1, 2, 3, and 4. They're depicted this way throughout the talk, and the identities of these proteins is not necessarily important for today's talk, but what is important is to understand that these are 4 naturally occurring proteins. They are from the same protein class, and they have distinct biological activities. And the goal for this project was to try to create a chimeric fusion of some combination of these 4 proteins that would have a novel activity that we could leverage. So I'll start by describing how I set up the cartridge screen. I broke it into a few different types of screens, the first of which was what I call the subunit portion of the screen. This is proteins 1, 2, 3, and 4, just individually cloned and expressed here. This serves as a nice control for us to understand how these proteins behave independently before we start sticking them together. The next part of the screen is what I call the linkers screen, which is the proteins 1, 2, 3, and 4 fused together with different amino acid linkers between each protein. You can see symbolized by the different symbols here. And then we have what I call the truncations portion of the screen. This is to say we took the constructs 1, 2, 3, and 4, and we truncated from the C terminus or from the N terminus. And then what I call the iterations part of the screen, which is different configurations of truncations and linkers. So here we have construct 14 looks the same as 16, but they have different linkers. 15 looks the same as 17, but they have different linkers, and so on. You get the idea. So I ordered all of those constructs from IDT, and I used the cloning kit from Nuclera to convert them into e-genes, and I used the Nuclera software to design my cartridge expression and purification screen. All of this, as Lucy and Lawrence have already described in detail for us. And this is what 192 data point expression data set looks like that comes off the cartridge. We have protein expression on the y-axis and all of the different constructs on the x-axis here. And I've put in this dashed line just to serve as a marker for us to judge what might be a poor expressor and what might be a strong expressor. And obviously looking at the data this way is not the most informative, so let me break it down into the different subsets of the data that we just described. Let's start with the subunit screen, and it turned out that the results of this screen were pretty clear trends across the rest of the data set as well. Here we have those 4 naturally occurring proteins, and we're just asking how well they express individually. And it turns out that protein number 1 expressed really well across the board, protein number 3 expressed pretty poorly across the board, and proteins 2 and 4 were somewhat in the middle. And we have a varying degree of variability dependent on the different expression conditions, but overall, 1 expressed well and 3 expressed pretty poorly. But what happens if we fuse construct 1 to construct 3? Well, fortunately, the capacity of the cartridge allowed us to ask that question as part of the same data set. We have included in the screen construct 22, which is a fusion of protein 1 and protein 3 here. And you can see that 22 looks a whole lot more like 3 than it does like 1, or we might say that the poor expression of construct 3 is dominant over the strong expression of construct 1. Now, this was a very valuable insight for us to get within the same data set, and it saved us a lot of time here not having to go back and ask this question in a follow-up experiment. It was just already available within the cartridge data. And here we have a couple more insights, or what I decided to call winning elements within the screen. If you remember the linker screen setup, this was proteins 1, 2, 3, and 4 with different amino acid linkers fusing them together. And it looks like for 4 out of the 5 constructs in the linker screen, we had protein that fell below our threshold of poor expression. And for one construct, we had strong expression, and that was this construct number 7. And the linker looks like this. And similarly, for the truncation screen, we had 4 constructs that fell below our threshold and 1 that was above the threshold. And maybe it's no surprise that construct number 11 has the fusion of protein 1 and 2, as we discussed that the inclusion of protein 3 tanks the expression of whatever it's attached to. And it might also be no surprise then that the overall winning construct from the cartridge screen was this construct number 14. And it has the winning elements of the subunit, the linker, and the truncation screen all put together. It is proteins 1 and 2 fused together with the winner of the linking screen and in the same configuration as the truncation screen had them. And so we found ourselves pretty hopeful that construct 14 might fit our need for that functional chimera we were out to identify, and so we decided to move it along to a scale-up. So that means we're moving away from the cartridge, and now we are working in a tube on the benchtop in a water bath, where we're going to conduct a 200 microliter scale-up. Now, when you conduct the cartridge screen, the instrument calculates and predicts what yield of a protein you might get from a 200 microliter scale-up. And so I'm plotting data here just to demonstrate that for each of the constructs we scaled up, we exceeded that predicted yield by quite a bit. So for constructs 1 and 2, this looked like we got a couple hundred micrograms of purified protein at the end. And for construct 14, we got almost 100 micrograms of purified protein. And I'll note that we're purifying constructs 1 and 2 in addition to 14 to serve as controls for our functional studies. And this is what the SDS page gels look like for the scale-up kit. Here we have input cell-free protein synthesis. You can see there's no band of our gene of interest. And then after overnight in the crude cell-free protein synthesis, we have a nice strong band showing expression of our gene of interest. And then this is the purity of the protein that we get after purification. And so this is more than enough protein at a high enough purity that we can move on to some functional studies. Now, for a functional analysis, we used biolayer interferometry, or BLI, to look at the binding of these different constructs to what would be naturally occurring ligands for constructs 1 and 2. So this is ligand A and ligand B, and we are hoping to engineer an enhanced affinity for this type of ligand for construct 14, and that looks like what we got. So here for ligand A, we see that construct 2 has a relatively low affinity, construct 1 has a relatively higher affinity, but then construct 14 has a much higher affinity than construct 1 or 2. And notably, it also has very low dissociation. And we see the same trend for ligand B that we saw for ligand A. We have a relatively small affinity for construct 2, relatively higher for construct 1, and then much greater for construct 14. And again, we see this characteristic relatively low level of dissociation. And so this looks like the novel activity, that enhanced activity that we were trying to engineer with this screen. And we are ready to do some kinetic analysis. So we were able to use that scale-up protein for construct 14 to do a nice dose response and calculate KDs. So you can see that data here, a nice dose response for ligand A and a nice dose response for ligand B. And for ligand A, we calculate a KD in the realm of 550 nanomolar. And for ligand B, that KD is 580 nanomolar. And so that's pretty stellar binding affinity to get out of this screen. Again, this is the E gene that we plugged into the cartridge. We just plugged it into a scale-up, and this is the performance of that protein. And so we're very happy with that, and we say that construct 14 is fit to advance for some further development. And so, in summary, this was a project that was really enabled by the eProtein discovery system. We really needed a high throughput approach, and we worked on this project sort of on and off over many months without much forward progress due to the limitations of our microbial and mammalian screening methods. And so the eProtein discovery really enabled us to make some forward progress on this project. The e-gene constructs were designed and screened within a few weeks. That's true. I designed them on the Nuclera software. They came in the mail a couple weeks later, and then I was able to quickly convert them to e-genes and put them onto the cartridge. After looking at the cartridge data, scaling up the protein was no sweat. You can get purified protein within a few days. I found that to be true. And the project timeline was months faster than previous approaches. I don't even want to know where we would be probably back at square one if the eProtein discovery had not enabled this project for us. And so with that, I will thank you for your time and attention, and I look forward to taking whatever questions you might have, and I'll hand the mic back to Lucy.
Thank you, Lawrence and Joel, for a very informative presentation. We have received several questions from our audience. This one looks like it's for IDT. What if the sequences are toxic or require special growth conditions? Really great question. Our SynBio Custom Solutions team is here to partner with you on projects that fall outside of our standard manufacturing. Whether you're working with special cell lines or special growth conditions, or even if you need longer genes and longer fragments, our scientists will work with you to build these tailored solutions to meet those specific project needs. We've seen various projects come in, and we're committed to helping you find the best path forward. Looks like the next question is, what if my project doesn't fit the catalog offerings that are on the webpage? I have a project where the fragment yields I need are much higher than that 1 microgram. So we've seen this come up frequently. It's a perfect example of where our SynBio Custom Solutions team can also step in. They work closely with customers to understand the project needs and design strategies that help achieve the desired yield without relying on overamplification, which can introduce unwanted mutations. And it looks like this next question is for Nuclera. What is the maximum size protein I can express on the eProtein discovery system?
Yeah, great. That's it's a great question. So we haven't tested the boundaries of protein size per se. However, proteins approaching 200 kilodaltons have actually been successfully expressed using eProtein discovery. And that's for soluble proteins, but for membrane proteins, we've tested large oligomeric proteins. So when using linear DNA fragments, size is typically limited to around 3,000 base pairs. But with our circular e-gene constructs, that can go up to 12,000 base pairs, which really opens the door for those larger proteins.
Thank you, Lawrence. Looks like there's another follow-up question here as well. Can you study protein complexes on the eProtein discovery system?
Yeah, another great question. So the easy answer is yes, the system can support protein complexes, but there's kind of two different strategies that can be deployed depending on whether the proteins are part of an obligate protein complex or not. So for non-obligate complexes, if the accessory protein is available, it can be supplemented as an additive into our cell-free system. And what that allows you to do is, your protein of interest can be expressed in the presence of the necessary folding or activity factors. But if co-expression is required, i.e. it is an obligate protein complex, we use a single detection system, and that means only the primary protein of interest will contain the detection and strep tags, while the co-expressed accessory protein will remain untagged. And the reason we do that is because it ensures that only the stable target protein is quantified, and that provides a really clear and accurate readout of protein quantity.
And what about the average yield and percentage purity?
Yeah. So as I alluded to in my talk, we're looking at microgram to milligram quantities from a scale up. Specifically, it's kind of about 150 micrograms per mil on average, and that purity is 92%. So that's really good, and it's suitable for many downstream applications, including your functional assays or structural studies.
Got it. And do the screening results correlate well to expression in cells?
Yeah. So we've observed over 80% correlation between our optimal constructs identified in our system compared to those performing best in E. coli. And so that's because tags or mutations that improve expression in our cell-free system often translate very nicely to improved yields in cells. And additionally, additives in screening can help identify ideal strains or media supplements to support proper folding and expression in vivo. One good example, which we've got a lot of information on, is, for example, if PDI GSSG is a top additive within your screen, you're likely to want to use Shuffle T7 as the bacterial strain. And that's because it contains, the necessary mutations to mimic that oxidative environment, and it expresses the protein disulfide
Got it. Thank you so much, Lawrence. And Joel, it looks like the next questions might be for Aldebaran. So for this project, did the protein yields from scale-up actions reflect the data from the cartilage?
Uh, yeah, definitely. So we only scaled up a handful of constructs from this screen that I showed today, but and we actually exceeded the predicted yield that the cartridge screen predicted. But we generally found uh, that the constructs scaled up um, in a manner that relatively matched the expression profile of those proteins in the cartridge. So protein one expressed really well in the cartridge. It also was the best expressor in the scale up, and the worst expressor on the cartridge was the worst expressor in the scale up. So I'd say that data matched pretty well.
And did the scale-up proteins plug directly into the VLI assay, or do you have to do other processes to move them along further?
This was pretty convenient for us. We had to do relatively minimal processing post scale up. So all we had to do was buffer exchange for the particular assay we were going to do, and then adjust the protein concentrations. And it was just a couple hours of work. So it was a pretty smooth transition from the scale up to the BLI assay for us.
Super exciting. Um, and then a question here is asking, can I request a custom protein project to run on the eProtein discovery system?
Great question. So I would say yes, reach out to our specialists on our website. Mention this webinar to them and tell them that you saw the screen on the eProtein discovery system, and you want to talk about whether it's the right approach for your project. So definitely give us a call, reach out.
Thank you so much. Unfortunately, we've run out of time to answer additional questions. We will contact you directly if you've asked a question and it was not answered here today. Thank you again for attending, and we wish you the best success in your research. And thank you, Lawrence and Joel. I appreciate it.
Thanks a lot.
Yeah, thanks, Lucy, for hosting.